Welcome everyone to this live podcast on Twitch, where you can find the news from the research industry that how our researchers keep on researching on various topics and domains to keep our lifestyle better, of course. And this article posted on our website named postscientist.com. If you are willing to check it out, please check it out. Otherwise, listening to this podcast will be enough for you guys as it acts like an archive on the daily basis, providing you the information from the research industry. So, without wasting any further time, let's get started with our first topic of the day. Building on more than 300 years of study, researchers developed a new way to understand bacterial growth. Okay, let's see. In labs, bacterial colonies usually grow in streak in petri dishes, but until now no one has known how the colonies organize themselves in more realistic three-dimensional 3D environments such as tissue and gels in our bodies or soils and sediments in the environment. This knowledge could be critical for advancing medical and environmental research. Now, a Princeton team has invented a new way to observe bacteria in 3D environments. They found that as the bacteria grow, their colonies constantly form fascinating rough shapes resembling a branch head of broccoli, much more integrate than forms typically seen in a flat dish. Ever since bacteria were discovered over 300 years ago, most lab research has studied them in a test tube or on petri dishes. Okay, said Sujit Datta, an assistant professor of chemical and biological engineering at Princeton and the study senior author. This was a result of practical limits rather than a lack of curiosity. If you try to watch bacteria grow in tissues or in soils, those are opaque and you can't actually see what the colony is doing. That has really been the challenge. Datta research team uncovered this behavior using a breakthrough experimental setup that allows them to make unprecedented observation of bacterial colonies in their natural three-dimensional state. Unexpectedly, the team found that the wild colonies' growth consistently resemble other natural phenomena like the growth of crystal or the spread of frost on a window pane. This kind of rough, branchy shapes are ubiquitous in nature, but typically in the context of growing or agglomerating non living systems, said Datta. What we found is that growing in 3D bacterial colonies exhibit a very similar process despite the fact that these are collectives of living organisms. The researcher presented this new description of how bacteria colonies grow in three dimensions in a paper published October 18 in the Proceedings of the National Academy of Science. Datta and his co-researchers hope the finding will assist with an area of research that deals with bacterial growth from the development of more effective antimicrobials to pharmaceutical, medical, and environmental research, as well as processes that harness bacteria for industrial use. At a fundamental level, we are excited that this work reveals surprising connection between the development of form and function, biological system, and studies of inanimate growth processes in materials science and statistical physics. But also, we think that this new view of when and where cells are growing in 3D will be of interest to anyone interested in bacterial growth, such as an environmental, industrial, and biomedical application, Datta said. For several years, Datta research team has been developing a system that allows them to analyze phenomena that are usually clogged in opaque settings, such as fluid flowing through soils. The team uses specially designed hydrogels, which are water absorbent polymers similar to those in gelatin and contact lenses, as matrices to support bacterial growth in 3D. Unlike those common versions of hydrogels, Datta's material are made up of extremely tiny balls of hydrogel that are easily deformed by the bacteria, allow for free passage of oxygen and nutrient that support bacterial growth and are transparent to light. It's like a ball pit where each ball is an individual hydrogel. They are microscopic, so you can't really see them, Datta said. The research team calibrated the hydrogel makeup to mimic the structure of soil or tissue. The hydrogel is strong enough to support the growing bacterial colony without presenting enough resistance to constrain the growth. As the bacterial colonies grow in the hydrogel matrix, they can easily rearrange the balls around them so they are not trapped, he said. It's like plunging your arm into the ball pit if you drag it through. The ball rearrange themselves around your arm. The researcher performed experiments with four different species of bacteria, including one that helps to generate kombucha tart taste to see how they grew in three dimensions. We change cell types, nutrient conditions, hydrogel properties. Datta said the researchers saw the same rough edge growth patterns in each case. We systematically changed all those parameters, but this appeared to be a generic phenomena. Datta said two factors seem to cause the broccoli shaped growth on a colony surface. First, bacteria with access to high levels of nutrients or oxygen will grow and reduce faster than ones in a less abductant environment. Even the most uniform environment has have some uneven density of nutrients, and these variation cause spots in the colony surface to surge ahead of or fall behind, repeated in three dimensions. This causes the bacteria colony to form bumps and nodules as some subgroups of bacteria grow more quickly than their neighbors. Second, the researcher observed that in three dimension growth, only the bacteria close to the colony surface grew and divided. The bacteria crammed into the center of the colony seemed to lapse into a dormant state because the bacteria on the inside were not growing and dividing. The outer surface was now subjected to pressure that would cause it to expand evenly. Instead, its expansion is primarily driven by growth along the very edge of the colony, and the growth along the edge is subject to nutrient variations that eventually result in bumpy, uneven growth. If the growth was uniform and there was no difference between the bacteria inside the colony and those on the periphery, it would be like filling a balloon, said Alejandra Martin Calvo, a postdoctoral research at Princeton and the paper first author. The pressure from the inside would fill in any permutation on the periphery. To explain why this pressure was not present, the researcher added a fluorescent tag to protein that become active in cells. When the bacteria grow, the fluorescent protein lights up when bacteria are active and remains dark when they are not. Observing the colonies, the researchers saw that the bacteria on the colony edge were bright green while the core remained dark. The colony essentially self organized into a core and a shell that behave in very different ways, Datta said. Nothing that the theory is that the bacteria on the colony edges scoop up most of the nutrient and oxygen, leaving little for the inside bacteria. We think they are going dormant because they are starved, Datta said, although he cautioned that further research was needed to explore this. Datta explained that the experimental mathematical models used by the researcher found that there was an upper limit to the bumps that formed on the colony surface. The bumpy surface is a result of random variation in the oxygen and nutrients in the environment, but the randomness tends to even out in certain limits. The roughness has an upper limit of how large it can grow. The floret size, if we are comparing it to broccoli, he said, we were able to predict that from the math, and it seemed to be an inevitable feature of large colony growing in 3D. Because the bacterial growth tended to follow a similar pattern as crystal growth, and other well studied phenomena of inanimate materials. Datta said that the researchers were able to adapt standard mathematical model to reflect the bacterial growth. He said future research will likely focus on better understanding the mechanism behind the growth, the implication of rough growth shapes of colony functioning, and applying these lessons to other areas of interest. Ultimately, this work gives us more tools to understand and eventually control how bacteria growth in nature, he said. So, I mean, it's a great thing, it's a great discovery for understanding the bacteria more by into the cells or growth of the cells. So, yep, it's going to be, I mean, it's going to be used in various application, of course, like uh, like in the 3D printing and many more other things which we I mean not coming up in my mind right now. So okay, moving on towards next topic. Physicists confirm H in proton structure. Okay, let's see. Nuclear physicists have confirmed that the current description of photon structure isn't all smooth sailing. A new precision measurement of the proton electric ability performed at the US Department of Energy Thomson Jefferson National Accelerator Facility has revealed a bump in the data in probes of the proton structure. Though widely thought to be a fluke when seen in earlier measurement, this new, more precise measurement has confirmed the presence of the anomaly and raises questions about its origin. The researcher has just published in the journal Nature. According to Ruonan Lee, first author on the new paper and a graduate student at Temple University, measurements of the proton electric polarizability reveal how susceptible the proton is to deformation or stretching in an electric field, like size or charge. Electric polarizability is a fundamental property of proton structure. What's more, a precision determination of the proton electric polarity polarizability can help bridge the different description of the proton depending on how it is probed. A proton may appear as a single particle or a composite particle made of three quarks held together by the strong phase. We want to understand the substructure of the proton, and we can imagine it like a model with three balanced quarks in the middle, Lee explained. Now put the proton in the electric field. The quarks have positive or negative charge. They will move in opposite directions, so the electric polarizability reflects how easily the proton will be distorted by the electric field. To probe this distortion, nuclear physicists used a process called a virtual Compton scattering. Start with a careful control beam of energetic electron from Jefferson Lab Continuous Electron Beam Accelerator Facility, a DOE Officer of Science User Facility. The electrons are sent crashing into protons. In virtual Compton scattering, electrons interact with the other particles by emitting an energetic photon or particle of light. The energy of the electron determines the energy of the photon it emits, which also determines how the photon interacts with other particles. Lower energy photons may bounce off the surface of the proton, while more energetic photons will blast inside the proton to interact with one of its quarks. Theory predicts that when these photon quark interactions are plotted at from lower to higher energies, they will form a smooth curve. Nikos Parvis, an associate professor of physics at Temple University and spokesperson for the experiment, said this simple picture didn't hold up to scrutiny. The measurement instead revealed an as yet unexplained bump. What we see is that there is some local enhancement to the magnitude of the polar stability. The polar stability decreases as the energy increases as expected, and at some point it appears to be coming temporarily up again before it will go down, he said. Based on our current theoretical understanding, it should follow a very simple behavior. Since we see something that deviates from the from this simple behavior, and this is the fact that is puzzling us at the moment. The theory predicts that the more energy electrons are more directly probing the strong force as it binds the quark together to make the proton. The weird spike in the stiffness that nuclear physicists how sorry have now confirmed in the proton quark signals that an unknown facet of the strong phase may be at work. There is something that we are clearly missing at this point. The proton is the only composite building block in nature that is stable. So if we are missing something fundamental there, it has implication or consequences for all of physics. Spartacus confirmed. The physicist said that the next step is to further tease out the details of the anomaly and conduct precision probes to check for other points of deviation and to provide more information about the anomaly source. We want to measure more points at various energy to present a clear picture and to see if there is any further structure there, Lee said. Sparvis agreed. We also need to measure precisely the shape of this enhancement. The shape is important to further elucidating the theory, he said. Of course, I mean, it's uh, it's the requirement of this to indicate or to be more to be to get more results from this research. So I mean, it's a requirement to precisely measure the shape of this announcement. Otherwise, it's not going to be further elucidated or further research goes on. So, yep. Moving on towards next topic. Once again, I mean, I have to cover lots of articles. So I don't able to provide you the short summary of the of every article. So please bear with me. And yes, uh, uh, the author itself, the article already described very well by the author. So I mean, there is no need for my explanation by my side. So yep, let's read it and cover it. Those articles. So yeah, meet the first Ninthal, Ninthal family. Ninthal family. Meet the first Ninthal family. It's a it's going to be an archaeology article. So let's read it and see. The first Neanderthal draft genome was published in 2010. Since then, researchers from the Max Planck Institute of Evolutionary Anthropology have sequenced a further 18 genomes from 14 different archaeological sites throughout Eurasia. While these genomes have provided insight into the broader strokes of Neanderthal history, we still know little of individual Neanderthal communities. To explore the social structure of Neanderthals, the researchers turned their attention to southern Siberia, a region that has previously been very fruitful for ancient DNA research, including the discovery of Denisovan homin remains at the famous Denisova cave. From work done at that site, we know that Neanderthals and Denisovans were present in this region over hundreds of thousands of years, and the Neanderthals and Denisovans have interacted with each other as the finding of a child with a Denisovan father and a Neanderthal mother has shown. Okay. First, Neanderthal community is their new study published in Nature. The researcher focused on the Neanderthal remains in the Chagra Sakya and Okla Tiknovo caves, which are within 100 kilometers of Denisov cave. Neanderthal briefly occupied these sites around 54,000 years ago, and multiple potentially contemporaneous Neanderthal remains had been recovered from their deposits. The researchers successfully retrieved DNA from 17 Neanderthal remains, the largest number of Neanderthal remains ever sequenced in a single study. Chagdskaya, Chagdskaya cave has been excavated over the last 14 years of research from the Institute of Archaeology and Anthropography, Russian Academy of Science. Besides several hundred thousand stone tools and animal bones, they also recovered more than 80 bone and earth fragments of Neanderthals, one of the largest assemblage of this fossil humans not only in the region but also in the world. The Neanderthals at Chekrovskaya and Oakland Dicknow hunted ibex, horses, bison, and other animals that migrated through the river valleys that the caves overlook. They collected raw material for their stone tools dozens of kilometers away, and the occurrence of the same raw material at both Chekrovskaya and Okoldenikov caves also support the genetic data that the groups inhabiting these localities were closely linked. Okay. Previous study of a fossil toe from Denisova cave showed that Neanderthals inhabited the Altai mountains considerably earlier as well, around 12,000 years ago, 1200,000 years ago. Genetic data shows, though, that the Neanderthals from Chagdskaya and Olkodnikov caves are not descendants of these earlier groups, but are closely related to European Neanderthals. This is also supported by the archaeology material. The stone tools from Chekrovskaya cave are most similar to the so-called Mikokuan culture from the Germany and Eastern Europe. The 17 remains came from the 13 Neanderthal individuals, 7 men and 6 women, of which 8 were adults and 5 were children and young adolescents. In their mitochondrial DNA, the researcher found several so-called heteroplasmids that were shared between individual. Heteroplasmids are a special kind of genetic variant that only persists for a smaller number of generations. The easternmost Neanderthals among these remains were those of a Neanderthal father and his teenage daughter. The researcher also found a pair of second-degree relatives, a young boy and an adult female, perhaps a cousin, aunt, or grandmother. The combination of heteroplasmids and related individuals strongly suggests that the Neanderthals in Chagdskaya cave must have lived and died at around the same time. Okay. Another striking finding is the extremely low genetic diversity within this Neanderthal community, consistent with a group size of 10 to 20 individuals. This is much lower than those recorded for any ancient or present day human community, and is more similar to the group size of endangered species at the verge of extinction. However, Neanderthals didn't live in completely isolated community. By comparing the genetic diversity on the Y chromosomes, which is inherited father to son, with the mitochondrial DNA diversity, which is inherited from mothers, the researcher could answer the question: was it the man or the woman who moved between communities? They found that the mitochondrial genetic diversity is much higher than the Y chromosome diversity, which suggests that the Neanderthal community were primarily linked by female migration despite the proximity to Denisova cave. These migrations do not appear to have involved Denisovans. The researcher found no evidence of Denisovan gene flow in the Chagdskaya Neanderthals in the last 20,000 years ago. before this individual lived. Okay. Our study provides a concrete picture of what a Neanderthal community may have looked like, says Benjamin Peter. The last order of the study, it makes Neanderthal seem much more human to me. Okay. I mean, I have much more information than that, but not able to provide you. Okay. Moving on to our next topic, number crunching math models may give policymakers major headache. Mathematical models that predict policy driving scenarios such as how a new pandemic might spread or the future amount of irrigation water needed worldwide may be too complex and delivering wrong answers. A new study reveals experts are using increasingly detailed models to better predict phenomena or gain more accurate insight in a range of key areas such as environmental climate change, climate sciences, hydrology, and epidemiology. But the pursuit of complex models as tools to produce more accurate projections and prediction may not deliver because more complicated models tend to produce more uncertain estimates. Researchers from the universities of Birmingham, Princeton, Reading, Barcelona, and Bergen published their findings today in Science Advances. They revealed that expanding model without checking how extra detail adds uncertainty limits the model usefulness as tool to inform policy decision in the real world. Arnold Pugh, Associate Professor in Social and Environmental Uncertainties at the University of Birmingham, commented as science keep on unfolding secrets. Models keep getting bigger, integrating now discovery to better reflect the world around us. We assume that more detailed models produce better prediction because they better match reality. And yet, pursuing ever complex models may not deliver the results we seek because adding new parameter brings new uncertainties into the model. These new uncertainty pile on top of the uncertainty already there at every model upgrade stage, making the model output fuzzier at every step of the way. This tendency to produce more accurate results affects any model without training or validation data used to check its output accuracy, affecting all global models such as those focused on climate change, hydrology, food production, and epidemiology, as well as the models projecting estimates into the future. Regardless of the scientific field, researchers recommend that the drive to produce increasingly detailed mathematical models as a mean to get sharper estimate should be reassessed. We suggest that modelers should calculate the model effective dimensions, the number of influential parameters, and their highest order interaction before making the model more complex. This allows to check how the addition of model complexity affects the uncertainty in the output. Such information is especially valuable for model aiming to play a role in policy making, added Dr. Pugh. Both modelers and policymakers benefit from understanding any uncertainty generated when a model is upgraded with novel mechanisms. Modelers tend not to submit their model to uncertainty and sensitivity analysis but keep on adding detail. Not many scholars are interested in running such an analysis on their model if it risks showing that the emperor runs naked and its alleged sharp estimates are just a mirage. Excess complexity prevents scholars and public alike to ponder the appropriateness of the model's assumptions, often highly questionable. Pugh and his team noted, for example, that global hydrological models assume that irrigation optimizes crop production water use, a premise at odds with practices of traditional irrigators. So these are the study, these are the findings given by our scientists and researchers. Moving on to our next topic, new research suggests our brains use quantum computation. Hmm. Scientists from Trinity College Dublin believe our brains could use quantum computation. Their discovery come after they adapted an idea developed to prove the existence of quantum gravity to explore the human brain and its workings. The brain function measured but also correlated to short-term memory, performance and consciousness awareness, suggesting processes are also a part of cognitive and conscious brain functions. If the team results can be confirmed, likely requiring advanced multidisciplinary approaches, they would enhance our general understanding of how the brain works and potentially how it can be maintained or even healed. They maybe also help find innovative technology and build even more advanced quantum computers. Dr. Christian Crenson, lead physicist at the Trinity College Institute of Neuroscience, Tensen, in the co-author of the research article that has just published in the Journal of Physics Communications. He said, "We adapted an idea developed for experiments to prove the existence of quantum gravity, whereby you take no known quantum systems which interact with an unknown system. If the known system entangled, when the unknown must be quantum system too. It circumvents the difficulties to find measuring devices for something we knew nothing about. For that experiment, we used proton spin of brain water as the known system. Brain water builds up naturally as fluid in our brains, and the proton spins can be measured using MRI, magnetic resonance imaging. Then, by using a specific MRI designed to seek entangled spins, we found MRI signals that resembles heartbeat evoked potentials, a form of EEG signals. EEG measured electrical brain currents, which some people may recognize from personal experience or simply from watching hospital dramas on TVs. Electrophysiological potential, like the heartbeat evoked potential, are normally not detectable with MRI, and the scientists believed they could only observe them because the nuclear proton spins in the brain were entangled. Dr. Kruskens added, if entanglement is the only possible explanation here, then that would mean that brain processes must have interacted with the nuclear spin, mediating the entanglement between the nuclear spin. As a result, we can deduce that those brain functions must be quantum. Because these brain functions were also correlated to short-term memory, performance, and conscious awareness, it is likely that those quantum processes are an important part of our cognitive and conscious brain functions. Quantum brain processes could explain why we still outperform supercomputers when it comes to unforeseen circumstances, decision making, or learning something new. Our experiment performed only 50 meters away from the lecture theater, where Schrodinger's presented his famous thoughts about life, may shed light on the mystery of biology and consciousness, which scientifically is even harder to grasp. Of course, I mean. Moving on towards next topic, methane eating bugs have been assimilating Earth microbes. In Star Trek, the bugs are a ruthless, high minded collective that assimilate other beings with the intent of taking over the galaxy. Here, on non fictional planet Earth, bugs are DNA packages that could help humans fight climate change. Last year, a team by Jill Benfield discovered DNA structure within a methane consuming microbe called methanopredans that appears to supercharge the organism metabolic rate. They named the genetic elements bugs because the DNA within them contains genes assimilated from many organisms. In a study published today as a cover item in Nature, the researcher described the curious collection of genes within bugs and began to investigate the roles these DNA packages play in environmental processes such as carbon cycling. First contact. Methanopredens are a type of archaea, unicellular organism that resemble bacteria but represent a distant branch of life that break down methane CH4 in soil, groundwater, and the atmosphere to support cellular metabolism. Methanopredens and other methane consuming microbes live in diverse ecosystems around the world but are believed to be less common than microbes that use photosynthesis, oxygen, or fermentation for energy. Yet, they play an outside role in Earth system processes by removing methane, the most potent greenhouse gas from the atmosphere. Methane traps 30 times more heat than carbon dioxide and is estimated to account for about 30% of human driven global warming. The gas is emitted naturally through geological processes and by methane generating archaea. However, industrial processes are releasing stored methane back into the atmosphere in varying quantities. Banfield, a faculty scientist at Lawrence Berkeley National Laboratory Berkeley Lab and professor of Earth and Planetary Science and Environmental Science, Policy and Management at UC Berkeley, studies how microbial activity shape large-scale environmental processes and how in turn environmental fluctuation alter the planet's microbiomes. As part of this work, she and her colleagues regularly sample microbes in different habitats to see what interesting genes microbes are using for survival and how these genes might affect global cycle of key elements such as carbon, nitrogen, sulfur. The team look at the genomes within cell as well as the portable packets of DNA known as extra chromosomal element ECE and that transfer genes between bacteria, archaea, and viruses. These elements allow microbes to quickly gain beneficial genes from the neighbors, including those that are only distantly related. While studying methano, methanopredens sampled from seasonal wetland pool soil is in California, the scientists found evidence of an entirely new type of ECE, unlike the circular strands of DNA that make up most plasmids. The most well-known type of extra chromosomal element, the new ECE are linear and very long, up to one third the length of entire methanopredens genome. After after analyzing additional samples from underground soil, aquifers, and riverbeds in California and Colorado that contain methane consuming archaea, the team uncovered a total of 19 distinct EC. They duped bogs. Using advanced genome analysis tool, the scientists determined that many of the sequences within the bog are similar to the methane metabolizing genes within the actual methanopredens genome. Some of the boards even encode all the necessary cellular machinery to eat methane on their own, as long as they are inside a cell that can express the genes. Imagine a single cell that has the ability to consume methane. Now you add genetic elements within that cell that can consume methane in parallel and also add genetic element that give the cell higher capacity. It basically creates a condition for methane consumption on steroids, if you will, explained co-author Kenneth Williams, a senior scientist and Banfield colleague in the Berkeley Lab Earth and Environmental Science area. Williams lead researcher at Raphael Colorado site where the best characterized bog was recovered and is also chief field scientist of a research site on the East River near Crested Butte, Colorado, where some of the Banfield's current sampling takes place. The East River Field site is a part of the Department of Energy Watershed Function Scientific Focus Area, a multidisciplinary research project led by Berkeley Lab that aims to link microbiology and biochemistry with hydrology and climate science. Our expertise is bringing together what are often thought of and treated as completely disparate fields of injury. Big science that links everything from genes all the way up to watershed and atmospheric processes. Resistant is fertile. Banfield and our fellow researcher at UC Berkeley Innovative Genomics Institute, including co-author and long-term collaborator Jennifer Don, Donna, hy- uh, hypothesize that the box could be residual fragments of entire microbes that were engulfed by methanopridans to aid metabolism, similar to how plant cells harnessed formerly free living photosynthetic microbes to gain what we call chloroplast, and how an ancient eukaryotic cell consumed the ancestor of today mitochondria. Eukaryotic cell consumed the ancestor of today mitochondria, of course. Based on this similarity in sequence, the engulfed cell could have been relative of methanopridans, but the overall diversity of gene found in bogs indicates that these DNA packages were assimilated from a wide range of organisms. No matter the origin, it is clear that bogs have existed alongside these archaea shuttling genes back and forth for a long time. Notably, some methanopridans were found with no bogs, and in addition to recognizable genes, the bogs also contain unique genes encoding other metabolic protein, membrane proteins, and extracellular proteins, all were certainly involved in electron conduction required for energy generation, as well as other proteins that have no unknown effects on their host. Until the scientists can culture methanopridans in a laboratory environment, they won't known for sure what capability the different bugs confer, why some microbes use them, and why others don't. One likely explanation is that bugs act as a storage locker for metabolic genes that are only needed at certain times. Ongoing methane monitoring research has shown that methane concentration can vary significantly throughout the year, usually peaking in the fall and dropping to the lowest level in early spring. The bugs therefore provide a competitive advantage to methane eating microbes like methanopridans during periods of abundance when there is more methane than their native cellular machinery can break down. Plasmids are known to serve a similar purpose, quickly spreading genes for resistance to toxic molecules like heavy metal and antibiotics, where the toxins are present in highly enough concentration to exert evolutionary pressure. There is evidence that different types of bugs sometimes coexist in the same host methanopridine cells. This opens the possibility that bugs could be spreading genes across lineages, said Banfield, broadly exploring the microbial universe. Since posting their article as a preprint last year, the team has begun follow up work to better understand how bugs may affect biological and geological processes. Some researchers are combining through data sets of genetic material from other microorganisms, looking for evidence that bugs exist in association with other species. While her colleagues are using lab based methods, co author Suzanne Mullen, a graduate student in Banfield Labs, will be getting her feet wet with some very picturesque field work. She recently started a project to sample microbes from the flood plains of the East River throughout the year to assess how seasonal changes in bog abductans and other microbes known to be involved in methane cycling correlate to seasonal flux of methane. According to the author, years down the line, carefully cultured microbes chock full of bogs could be used to reduce methane and curb global warming. It's all to benefit the collective life on Earth. Sorry, guys. Sorry, guys. I mean, they deserve it, so yep. Moving on to our next topic. Genetic analysis of bones found in Greenland helps explain how early people use of resources helped them to survive. A team of researchers affiliated with several institutions in Denmark and Greenland, working with another colleague from Australia, has found that early human living in Greenland ate a much more varied diet than previously believed. The study is published in Nature Human Behavior. Scientists studying the history of Greenland have often wondered how early humans could have survived in such a cold climate. Prior research has shown that there have been four major migrations to Greenland: the Sikyuk, the Dorset, the Norse, and the Thule. Only the Thule become permanent residents. The first group, the Sikyuk people, arrived approximately 4,500 years ago and remained in Greenland until a strong cooling period drove them away after 1700 years. Prior research efforts have suggested they ate fish, seals, and perhaps even sometimes of whales, despite the limited toolset. In this new effort, the researcher took a closer look at the diet of the Sikyuk and Sakyek and the diets of their other groups that followed them. Their work evolved conducting a DNA analysis of bone fragments collected from kitchen middens, a term used in Greenland to describe mounds of bones left behind by humans as they were discarded after meals. In studying approximately 2,500 bone shards, and the researcher found they were able to identify 42 species of creatures eaten by early humans, many of which came as a surprise. The team found, for example, DNA from extinct species of reindeer, one that was much smaller than those that live in Greenland today. They also found evidence of multiple types of whales, fin, sperm, narwhal, and bowhead. Bowhead were the most prevalent, which the researcher noted makes sense because they are relatively easy to kill. In all, the researcher found evidence of 20 mammal species, 9 kinds of fish, including a surprising number that were quite small, suggesting the use of nets, and 13 types of birds. The DNA evidence revealed not just the type of creature that were captured and eaten by early people and the researcher note, but also sheds light on the technical know how of those who succeeded in hunting them. Oh, okay, okay. Moving on to our next topic, the secret behind spectacular blooms in the world driest desert. Okay. The Atacama Desert, which stretches for approximately 1600 kilometers along the western coast of the cone of South America, is the driest place on Earth. Some weather station there have never recorded rainfall throughout their existence, but it's far from barren. Many species live here than that occur now where else adapted to its extreme conditions, and approximately every 5 to 10 years from September to mid-November, the Atacama hosts one of the most spectacular sights of the natural world, the Desert of Florido, literally blooming desert. The mass blooms, one of which is currently going on in the northern Atacama after abundant rainfall earlier this year, often attract media attention from around the globe, but which psychological and evolutionary mechanism enable the great diversity of colors, shapes, and visual patterns of flowers during Desideratus floridos, and how do pollinators in the Atacama, mainly patrons like solitary wasps and bees, for whose benefit this visual Ganza evolved, perceive all this variation. That's the subject of a new study in Frontiers in Ecology and Evolution. Our aim was to shed light on the ecological and evolution mechanism that cause biological diversity in extreme environments like the Atacama Desert, said first author Dr. Jamie Martin Holmes, a researcher at the Institute of Agricultural Research, La Cruz, Chile. Here we showed that flowers of the pussy paw, Crichton longi scarpa, a representative species from district of Floridos in the Atacama Desert, are highly variable in the color and patterns they present to pollinators. This variability the probably result from different so-called battalion pigments in the flower petals. Model species. Martin, Holmes, and colleagues studied at the University of Florido event in late 2021 near the city of Cladera in northern Chile. Despite being smaller in size than the event currently going on, it was clearly visible to satellites. A dominant species was C. longiscapa family Montiana K, an annual plant up to 20 centimeter high, which bloomed in two distant patches, tens of kilometers across. These patches are considered off to humans uniformly purple and yellow flowers. Between them grew numerous intermediate, that is reddish, pinkish, and white flowers of the same species, strongly suggesting that the purple and yellow morph are heritable variants that can interbreed. Visualizing flowers can, as insects sees them, insects with their compound eyes and different sensitivities see the world very differently than we do. For example, most patrons have three types of photoreceptors which are maximally sensitive to UV, blue, and green. Marks and Holmes at Yale use cameras sensitive to visual light and UV and spectrometers to measure the refraction, absorption, and transmission of different wavelengths by the petals of a total of 100 purple, yellow, red, pink, and white C. longiscapa of flowers. This enabled them to produce composite images of this variant are seen by their many species of pollinator diversity hidden from human eyes. The result showed that just within this single plant species, the diversity pers- uh, perceptible to pollinators was greater than to us. For example, hymenopterians, just like us, can easily distinguish between red, purple, white, and yellow variants, but they can also distinguish between flowers with a high versus of low UV reflection. Among yellow and purple flowers, a UV bull's eye pattern at the heart of some flowers, which guides pollinator to the nectar and pollen, is invincible to us. Hmm. An exception are UV reflecting pink and reddish C. longiscapa, which are quite distinct to human eyes but probably appear similar to hymenopterans. This visual diversity of C. longiscapa flowers is probably mainly due to differences between bitlanes, yellow, orange, and purple pigments that are typical trait of the plant order Cryophlalius, to which the pussy paws belong. Bitlanes don't just give colors to flowers, they also protect them from drought, salt, stress, and damage from reactive oxygen radicals under environmental stress. Traits highly beneficial in deserts. Pollinator derived selection for new variants. The author hypothesized that are observed standing diversity within C. Long scarp of flowers in driven by difference in the sensitivity and preference for different colors and pattern across many species of pollinator, an evolutionary experiment going on right now which mostly escapes our eyesight. The great variation in flower color with the C. long scarpa can be explained if different species of pollinating insects through their preference for particular flowers, colors, and patterns could cause this variant to become reproductively isolated from other individuals of the same plant species. This ongoing process could ultimately lead to the origin of new races or species, said Martins Holmes in our next studies, which will further investigate the chemical identity and the biological synthesis pathways of beetles and other flower pigments, as well as their relationship to traits such as the scents produced by the flowers. This should help us to understand their role in shaping the interactions between plants and their pollinators and in the plant tolerance to biotic and abiotic stressors under fluctuating climate conditions, said Marnett Holmes. Okay. Moving on to our next topic. Sickly shades of grey disease outbreaks influence the color of walls across North America. New research from the University of Oxford, Yellowstone National Park, and the Penn State, published today in the journal Science, may have finally solved why walls change color across the North American continent. If you were to travel from Arctic Canada and head south down the Rocky Mountains into the US toward Mexico, the further south you go, the more black walls there are. The reason why have long puzzled scientist Professor Tim Carlson from the Department of Biology, University of Oxford, who led the work, explains in most parts of the world, black walls are absent or very rare, yet in North America they are common in some areas and absent in others. Scientists have long wondered why. We now have an explanation based on wall surveys across North America and modeling motivated by extraordinary data collected by co-authors who work in Yellowstone. Coat colored in walls, Canis lupus, is determined by a gene called CPD103. Depending on the variant of the gene a wolf has, co- its cost sorry its coat can either be black or gray. The researcher postulated that this gene also play a role in protecting against respiratory diseases such as canine dysentery virus CDV. This is because the DNA region containing the gene also encodes for a protein that plays a role in defending against infection in the lungs of mammals. They predicted that having a black coat would be associated with the ability of whales to survive an infection with CDV. To test this idea, they analyzed 12 wolf populations from North America to examine whether the probability of a wolf being black was predicted by the presence of CDV antibodies. If a wolf has CDV antibodies, then it has caught CDV in the past and survived. They found that wolves with CDV antibodies were more likely to be black than gray. They also found that black wolves were more common in areas where CDV outbreaks occurred. The researcher analyzed over 20 years worth of data from the wolf population at Yellowstone National Park. They found that black wolves were more likely to survive CDV outbreaks compared with gray wolves. This result led them to hypothesize that in areas where distempered outbreaks occurred, wolves could choose mates of the opposite color to maximize the change. Their cubes would have black coats. They used a simple mathematical model to test this idea. Excitingly, The prediction from their model closely matched the observation that black and gray walls were more likely to appear in the areas where CDV outbreaks are common. The competitive advantage is lost in areas where CDV outbreaks do not occur. These results are consistent with the idea that the frequency of CDV outbreak across North America is responsible for the distribution of black walls because having the gene of for a black coat may have provided protection against the virus. It also explains why mating pairs in Yellowstone, where canine distemper outbreaks occur, tend to be black-gray. Peter Hunson, William, professor of biology, Penn State, said it's intriguing that the gene of protection against CDV came from domestic dogs brought by the first humans entering North America, and the CDV disease virus emerged in North America many thousands of years later, once again from dogs. What I love about this study is how we have been able to bring together experts from so many fields and a range of approaches to show how diseases can have remarkable impacts on wolf morphology and behavior. We are learning that disease is a major evolutionary driver that impacts so many aspects of animal populations. The researchers speculate that other species may follow a similar pattern to wolves. Many amphibians, birds, and non-human mammals have association between color and disease resistance. It might be that the presence of disease or how frequently a disease outbreak occurs is an important factor affecting the color of meat an animal prefers. Okay. Moving on to our next topic. Scientists map key protein structure of hepatitis virus. Hepatitis C virus. Okay. A team led by scientists at Script Research and the University of Amsterdam has achieved an important goal in virology, mapping at high resolution critical protein that stirred the surface of hepatitis C virus HCV and enable it to enter host cells. The discovery reported in Science on October 21st, 2022 details key sites of vulnerability on the virus sites that can now be targeted effectively with vaccines. This long sought after structural information on HCV puts a wealth of previous observations into a structural context and pave the way of rational vaccine design against this incredible difficult target, says study co-senior author Andrew Ward, PhD professor in the Department of Integrative Structural and Computational Biology at Scripps Research. The study was the product of a multi-layer collaboration that included the Ward Laboratory, the lab of Gabriel Lander, PhD, also professor in the Department of Integrative Structural and Computational Biology at Scripps Research, the lab of Roger Sanders, PhD, of the University of Amsterdam, and the lab of Max Crispin D. Phil, and the University of Southampton. It is projected that roughly 60 million years, millions people globally, including about 2 million Americans, have chronic HCV infections. The virus infects liver cells, typically establishing a silent infection for decades until liver damage becomes severe enough to cause symptoms. It is a leading cause of chronic liver disease, liver transplants, and primary liver cancers. The origin of the virus are uncertain, but it is thought to have emerged at least several hundred years ago and then eventually spread globally, especially via blood transfusions in the later half of the 20th century. While the virus was mostly eliminated from blood banks after its initial discovery in 1989, its continues to spread chiefly via needle sharing among intravenous drug users in developed countries and by the use of sterilize medical instruments. In developing countries, the leading HCV antiviral drugs are effective but far too expensive for large scale treatment. An effective vaccine could eventually eliminate HCV as a public health burden. However, no such vaccine has ever been developed, largely because of the extraordinary difficulty in the studying HCV envelope protein complex, which is made of two viral proteins called E1 and E2. The E1 and E2 complex is very flimsy and it's like a bag of wet spaghetti, also changing shape, and that's why it's being extremely challenging to image at high resolution, says co-first author Lisa Anson Wilson, PhD, a postdoctoral research associate in both the Lander and Ward labs at Script Research. In the study, the researcher found that they could use a combination of three broadly neutralizing anti-HCV antibodies to stabilize the E1 E2 complex in a natural conformation. Broadly neutralizing antibodies are those that are able to protect against a broad range of viral strains by binding to relatively non-varying sites on the virus in ways that interrupt the viral life cycle. The researcher imaged the antibody stabilized protein complex using low temperature electron microscopy. With the help of advanced image analysis software, the researchers were able to generate as E1 and E2 structural map of unprecedented clarity and extent at near atomic scale. Resolution details included most of the E1 and E2 protein structures, including the key E1 E2 interface and three antibiotic binding sites. The structural data also eliminated the thicket of sugar related glycan molecules atop. Even E2 viruses often use glycans to shield themselves from the immune system of an infected host, but in the case the structural data showed that HCV glycans apparently have another key role in helping to hold the flimsy even E2 complex together. Having this detail of even E2 will help researchers rationally design a vaccine that powerfully elicits these antibodies to block HCV infection. The structural data are also should allow us to discover the mechanism by which these antibodies neutralize HCV, says co-first author Alba Torrents de la Pena, PhD, a postdoctoral researcher in the World Lab. Okay, moving on to our next topic, completing Einstein homework on special relativity in electromagnetism. Okay, interesting. Albert Einstein, one of the most celebrated modern scientists, proposed the revolutionary theory of special relativity more than a century ago. This theory forms the basis of most what we understood about the universe, but a part of it has not been experimentally demonstrated until now. Scientists from the Institute of Laser Engineering at Osaka University have used ultrafast electro-optic measurement to visualize for the first time the contraction of the electric field around an electron beam moving at closer to the speed of light and demonstrate the generation process. In the theory of special relativity, Einstein proposed that in order to properly describe the motion of objects moving past an observer at close to the speed of light, one need to employ a Lorentz transformation that mixes the coordinates of space and time. He was able to explain how this transformation made the equation for electric and magnetic fields self-consistent. While various effects of relativity have been demonstrated many times to a very high degree of experimental precision, there are still aspects that remain unrevealed in experiments. Ironically, this includes the contraction of the electric field represented as a phenomenon of special relativity in electromagnetism. Now, the researcher team at Osaka University has demonstrated this effect experimentally for the first time. They accomplished this feat by measuring the profile of the Coulomb field in the space and time around a high energy electron beam generated by linear particle accelerator. Using ultra-fast electro-optic sampling, they were able to record the electric field with extremely high temporal resolution. It has been reported that the Lorentz transformation of time and space, as well as those of energy and momentum, were demonstrated by time dilation experiments and rest mass energy experiments, respectively. Here, the team looked at a similar relativistic effect called electric field contraction, which corresponds to the Lorentz transformation of electromagnetic potential. We visualize the contraction of electric field around an electron beam propagating close to the speed of light, says Professor Makoto Nakajima, the project leader. In addition, the team observed the process of electric field contraction right after the electron beam passed through a metal boundary. When developing the theory of relativity, it is said that Einstein used through experiments to imagine what it would be like to ride on a wave of light. There is something poetic about demonstrating the relativistic effects of electric field more than 100 years after Einstein predicted it, says Professor Nakashima. Electric field were a crucial element in the formation of the theory of relativity in the first place. This research with observation matching closely to Einstein's prediction of special relativity in electromagnetism can serve as a platform for measurements of energetic particle beams and other experiments in high energy physics. The paper is published in Nature Physics. Okay. Moving on to our next topic, tapping hidden visual information and all in one detector for thousands of colors. Okay. Spectrometers are widely used throughout industry and research to detect and analyze light. Spectrometer measure the spectrum of light, its strength at different wavelengths, like the colors in a rainbow, and are an essential tool for identifying and analyzing specimens and materials. Integrated on-chip spectrometers would be a would be of great benefit to a variety of technologies, including quality inspection platform, security sensor, biomedical analyzers, healthcare system, environmental monitoring tools, and space telescope. An international research team led by researchers at Aalto University have developed high sensitivity spectrometers with high wavelength accuracy, high spectral resolution, and the broad operation bandwidth using only a single microchip sized detector. The research behind the new ultra mini Anodized spectrometer was published today in the journal Science. Our single detector spectrometer is an all-in-one device. We designed this electronic lab on a chip with artificial intelligence replacing conventional hardware such as optical and mechanical components. Therefore, our Therefore, our computation spectrometer doesn't require separate bulky component or an added design to disperse and filter light. It can achieve a high resolution comparable to benchtop system, but in a much smaller package, says postdoctoral research Han Yoon. With our spectrometer, we can measure light intensity at each wavelength beyond the visible spectrum using a device at our fingertips. The device is entirely electrical controllable, so it has enormous potential for scalability and integration. Integrating it directly into a portable device such as smartphones and drones could advance our daily lives. Imagine that the next generation of our smartphones cameras could be fitted with the hyperspectral cameras that outperform color cameras, he adds. Shrinking computation spectrometers is essential for their use in chips and implantable application. Professor Jinping Sun, the head of the research team, says Conventional spectrometers are bulky because they need optical and mechanical components, so their on-chip applications are limited. There is an emerging demand in this field to improve the performance and usability of spectrometers. From this point of view, miniaturized spectrometers are very important to offer high performance and new function in all fields of science and industry. Professor Pertti Hankanen adds that Finland and Aalto have invested in photonics research in recent years. For example, there has been great support from the Academy of Finland Center of Excellence on Quantum Technology, flagship on photonics research and innovation Institute Q, and the Otano infrastructure. Our new spectrometer is a clear demonstration of the success of these collaborative efforts. I believe that with further improvements in resolution and efficiency, these spectrometers could provide new tools for quantum information processing. Moving on to our next topic, researchers discovered previously unknown mineral- mineralogy of the deep earth. Okay. What is the structure of the earth? For starters, it consists of several layers: the crust, the upper and lower mantle, and the core. The mantle makes up most of our planet volume, 84%, and the lower mantle represents 55% of Earth volume. It is also hotter and denser than the upper mantle. The lower mantle has played an important role in the Earth evolution, including how Earth has cooled over billions of years, how material have been circulated, and how water is stored and transported from to the deep interior over a geological timescale. For more than 7 decades, the mineralogy of the lower mantle has been studied extensively. The decades of study, including laboratory experiment, computational simulation, and the study of inclusion in deep demands, led to the conclusion that the lower mantle consists of three main minerals: bridge manite, ferro and deviomyota. In a study recently published in Nature, a team of scientists, including Ben Guan Kyo, former PhD student at ASU, now a postdoctoral research at Michigan State University, and Sang Hyun Dang Shim, professor at the School of Earth and Science Exploration, and now Drusky, professor of material research at ASU, have completed a new high pressure experiment employing some different style of heating to reveal an additional mineral residing in the lower mantle. Among these three main minerals, two mineral bridge magnite and deviomyota have both a so-called perovskite type crystal structure. The structure is also widely known in physics, of course, chemistry, and material engineering, as some material with the perovskite type structure have shown superconductivity. At shallow depths, mineral with similar crystal structures often merge and become single minerals, typically under a high temperature environment. For example, mineral diopside has both calcium and magnesium and is stable in the crust. Despite the structural similarity, however, existing studies have shown that Deviomyota, rich in calcium, and bridge magnite, enriched in magnesium, remain separate throughout the lower mantle. While diomyota and bridge magnite do not merge to one, despite the fact that they have very similar atomic scale structure. The question has fascinated researchers over 2 decades. Say Shem, many attempts have been made to find conditions where these two minerals merge, yet the answer from experiment has been constantly two separate minerals. This what we felt we need more fresh new ideas in experiments. The new experiment was an opportunity for the research group to try various heating techniques to compare method instead of increasing temperature slowly in conventional high pressure experiments. They increase temperature very fast to the high temperature related to the lower mantle reaching 3000 to 3500 Fahrenheit within a second. The reason for this was that once a two perovskite structured minerals form, it becomes very difficult for them to merge even if they enter into temperature conditions where a single perovskite mineral should be stable. By heating the sample fast to target temperature, Co and Shem were able to avoid formation of two perovskite structured minerals at lower temperatures. Once they reached the temperature at the lower mantle, they monitored what minerals form for 50 to 30 minutes using X-ray beams at the advanced photon source. They found that only single perovskite mineral forms unexpected from the previous experiments. They found that at sufficiently high temperature greater than 3500 Fahrenheit, Davium iota and bridge magnetite become a single mineral in the perovskite type structure. It has been believed that a large size difference between calcium and magnesium, the major uh, cations, and of Davium iota and bridge magnetite, respectively, should hinder this two material from merging, said Co. But our study shows that they can overcome such difference in hot environments. Uh, I mean, I will be back in a minute, guys. The experiment suggests that the deeper lower mantle with a sufficiently high temperature should have mineralogy different from the shallower lower mantle because the mantle was much warmer in early Earth. The group new result indicate that most of the lower mantle had a single perovskite structured mineral then, which means the mineralogy differed from the present day lower mantle. This new observation has a range of substantial impacts on our understanding of the deep earth. Many seismic observations have shown that the deeper lower mantle properties are different from the shallow lower mantle. The changes are reported to be gradual. This merge of bridge magnetite and Davium iota is shown to be gradual in the research group. Experiments also the property of a rock with three main minerals: bridge magnetite, ferro, periclase, and Davium iota does not match well with the properties of the deeper lower mantle. Co and collaborator predict that these unresolved problems can be explained by merge of bridge magnetite and Davium iota to a single perovskite structure mineral. I mean, great man, great. They deserve it. It deserve it. Okay, moving on to our next topic. Stem cell study reveals how neurons from PTSD patient react to stress. Stem cell derived neurons from combat veterans with post traumatic stress disorder (PTSD) react differently to a stress hormone than those from veterans without PTSD, a finding that could provide insights into how genetics can make someone more susceptible to developing PTSD following trauma exposure. The study published October 20 in Nature Neuroscience is the first to use induced pluripotent stem cell models to study PTSD. It was conducted by a team of scientists from the Akian School of Medicine at Mount Sinai, the James G. Peters Veterans Affairs Medical Center, and the Yale School of Medicine and the New Work Stem Cell Foundation Research Institute, New York. Yes. Post traumatic stress disorder can develop following severe trauma and is an enormous public health problem for both veterans and civilians. However, the extent to which genetic and environmental factors contribute to individual clinical outcomes remains unknown. To bridge this information gap, the research team studied a cohort of 39 combat veterans with and without PTSD, who were recruited from the James J. Peters Veterans Affairs Medical Center in the Bronx. Veterans underwent skin biopsies, and their skin cells were reprogrammed into induced pluripotent stem cells. Reprogramming cells into induced pluripotent stem cells is like virtually taking cells back in the time to when they were embryonic and had the ability to generate all the cells of the body, said Rachel Yehuda, PhD professor of psychiatry and neuroscience and at Yankian Mount Sinai, director of mental health for the James J. Peters Veterans Affairs Medical Center and the senior author of the paper. These cells can then be differentiated into neurons with the same properties as the person brain cell had before trauma occurred to change the way they function. The gene expression networks from these neurons reflect early gene activity resulting from genetic and very early developmental contributions, so they are a reflection of the pre-combat or pre-trauma gene expression state. Two people can experience the same trauma, but they won't necessarily both develop PTSD, explained Crimson Brennan, PhD Elizabeth Mears and House Jameson, professor of psychiatry at Yale University of Medicine and New York New York Stem Cell Foundation Research Institute. Robertson investigator Lumina, who co-lead the study. This type of modeling brain cell from people with and without PTSD helps explain how genetics can make someone more susceptible to PTSD. To mimic the stress response that triggers PTSD, the scientists exposed the induced pluripotent stem cell-derived neurons to the stress hormone hydrocortisone, a synthetic version of the body own cortisol that is used as part of the flight response. The addition of stress hormones to these cells stimulates biological effects of combat, which allow us to determine how different gene networks mobilize in response to stress exposure in brain cell, explained Dr. Yehuda. Using gene expression profiling and imaging, the scientists found that neurons from individuals with PTSD were hypersensitive to this pharmacological trigger. The scientists also were able to identify the specific gene networks that respond differently following exposure to the stress hormones inside the cell of PTSD affected individual. More similar studies of PTSD to date have used blood sample from patients, but since PTSD is rooted in the brain, scientists need a way to capture how the neurons of an individual prone to the disorder are affected by stress. Therefore, the team opted to use stem cells as they are uniquely occupied to provide a patient a specific, non-invasive window into the brain. You can't easily reach into a living person brain and pull out cells, so stem cells are our best way to examine how neurons are behaving in a patient. Of course, said Dr. Brennan. Uh, I mean, okay. NINYSCF, New York Cell Foundation. Scientists use the scalable automated robotic system, the NISCF Global Stem Cell Array, to create a stem cell and then glutamatergic neurons from patients with PTSD. Glutamatergic neurons help the brain send anxiety signals and have previously been implicated in PTSD. As this was the first study using stem cell models of PTSD, it was important to study a large number of individuals, said Daniel Paul, PhD, NYSCF, Senior Vice President, Discovery and Platform Development, who co-lead the study. At the scale of this study, automation is essential. With that, we can make standardized cells that allow for meaningful comparison between numerous individuals, pointing to key differences that could be critical for discovering new treatments. Leveraging the hallmarks of stress PTSD cells for new treatment, the team gene expression analysis revealed a set of genes that were particularly active in the PTSD prone neurons following their exposure to stress hormones. Importantly, the gene signature we found in the neurons was also apparent in brain sample from decreased individual with PTSD, which tell us that stem cell models are providing a pretty accurate reflection of what happens in the brain of living patients, noted Dr. Paul. Moreover, the distinctions between how PTSD and non-PTSD cells responded to stress could be informative in predicting which individuals are at high risk for PTSD. What's really exciting about our findings and the opportunity they offer for accelerating the diagnosis and the treatment of PTSD, Dr. Paul continued. Importantly, having a robust stem cell model provides an ideal avenue to drug screening in the dish, even across diverse patient population. We are working on finding already approved drugs that could reverse the hypersensitivity we are seeing in neurons. Added Dr. Brennan, that way any drugs we discover will have the fastest possible path to helping patients. The researcher plan to continue leveraging their induced pluripotent stem cell models to further investigate the genetic risk factor pinpointed by this study and the study how PTSD affects other types of brain cells, helping to broaden opportunities for therapeutic discovery. NYSCF is incredibly proud to have generated the first ever induced pluripotent stem cell models from the individual with PTSD as a part of the landmark study in partnership with the world class scientists, said NYSCF Interim CEO Derek Rossi, PhD. The collaborative work underscores and the unique value of stem cell modeling for studying and demystifying challenging diseases and for discovering innovative strategies that could be lead to urgently needed treatments. Hmm. Okay, moving on to our next topic and the last topic of the podcast. So, okay. Deep learning with light components of machine learning model encoded onto light waves. Ask ask a smart home device for the weather forecast, and it takes several seconds for the device to respond. One reason this latency occurs is because connected devices don't have enough memory or power to store and run the enormous machine learning models needed for the device to understand what a user is asking for of it. The model is stored in a data center that may be hundreds of miles away, where the answer is computed and sent to the devices. MIT researcher has created a new method for computing directly on these devices, which drastically reduces this latency. Their technique shifts the memory intensive steps of running a machine learning model to a central server where components of the model are encoded onto light waves. The waves are transmitted to a connected device using fiber optics, which enable tons of data to be sent lightning fast through a network. The receiver then employs a simple optical device that rapidly performs computation using the parts of a model carried by those light waves. This technique leads to more than a hundredfold improvement in energy efficiency when compared to other models. It could also improve security since user data do not need to be transferred to a central location for computation. This method could enable a self-driving car to make decisions real time while using just a tiny percentage of the energy currently required by power hungry computers. It could also allow a user to have a latency free conversation with their smart home device we use for live video processing over cellular networks, or even enable high speed image classification on a spacecraft millions of miles from Earth. Every time you want to run on a neural network, you have to run the program, and how fast you can run the program depends on how fast you can pipe the program in from memory. Of course, our pipe is massive. It's correspond to sending a full feature length movie over the internet every millisecond or so. Okay, that is how fast data comes into our system, and it can compute as fast as that, says senior author Trick England, and as associate professor in the Department of Electrical Engineering and Computer Science (EECS) and a member of the MIT Research Laboratory of Electronics. Joining England on the paper is lead author of EECS, a grad student Alexander Slutz, EECS grad student Samuel Bendy, Bendy Padhai, research scientist Ran Hamley, as well as others from MIT and the MIT Lincoln Laboratory and the Nokia Corporation. Okay. The research will be published in Science, lightening the load, neural networks, or machine learning models that use layer of connected nodes or neurons to recognize the pattern in data sets and perform tasks like classifying images or recognizing speech. But these models can obtain billions of weight parameters, which are numeric values that transform input data as they are processed. These weights must be stored in memory. At the same time, the data transformation process involves billions of algebraic computations, which require a great deal of power to perform. The process of fetching data, the weights of the neural network in this case, from memory and moving them to parts of a computer that do the actual computation is one of the biggest limiting factors to speed and energy efficiency, says Sklutz. So our thought was, why don't we take all that heavy lifting, the process of fetching billion of weights from memory, move it away from the edge device and put it someplace where we have abducted access to power and memory, which gives us the ability to fetch those weights quickly, he says. The neural network architecture they developed, net, uh, netcast, involves storing weights in a central server that is connected to a novel piece of hardware called a smart um, transceiver. The smart transceiver, a thumb sized chip that can receive and transmit data, uses technology known as silicon photonics to fetch trillions of weight from memory each second. It receives weights as electrical signal and imprints them onto light waves. Since the weight data are encoded as bits, ones and zeros, the transceiver converts them by switching laser. The laser is turned on for a 1 and off for a 0. It combines these light waves and then periodically transfers them through a fiber optic network, so a client device doesn't need to query the server to receive them. Optic is a great Optic is great because there are many ways to carry data with optic. For instance, you can put data on different colors of light, and that enables a much higher data throughput and a greater bandwidth than with electronic explain Bandyopadhyay trillions per second. Trillions per second. Once the light waves arrive to the client device, trillions per second. Trillions per second. Once the light waves arrive at the client device, a simple optical component known as broadband max standard modulator use them to perform superfast analog computation. This involves encoding input data from the device, such as sensor information, onto the weights. Then it sends each individual wavelength to the receiver that detects the light and measure the result of the computation. The researcher device a way to use this modulator to do trillions of multiplication per second, which vastly increase the speed of computation on the device, which using only a tiny amount of power. In order to make something faster, you need to make it more efficient. But there is a trade-off. We have built a system that can operate with about a milliwatt of power, but still do trillions of multiplication per second in terms of both speed and energy efficiency. That is a gain of orders of magnitude. Slot says they tested this architecture by sending weights over an 86-kilometer fiber that connects their lab to MIT Lincoln Laboratory. Netcast enabled machine learning with high frequency 98.7% for image classification and 98.98% for digital recognition at rapid speeds. We had to do some calibration, but I was surprised by how little work we had to do to achieve some high frequency out of the box. We were able to get commercially relevant accuracy adds hammerly. Moving forward, the researcher want to iterate on the smart transceiver chip to achieve even better performance. They also want to miniaturize the receiver, which is currently the size of shoebox, down to the size of a single chip, so it could fit onto a smart device like a cell phone. I mean, yep. Uh, uh, as I previously discussed in the many of the podcasts, that we have started a newsletter providing the information about which is not easily available on the surface web. So please subscribe to this newsletter. And yes, I mean, to sustain this business model, it's necessary to have to attain those things. So please, it's a request. Otherwise, it's up to you, of course. And at the last, I just wanna say, you guys, stay curious. Sorry, keep researching. Stay curious. Stay healthy.